Welcome back viewers to my channel. In this video we will be discussing about peptides and peptide bond as well as Ramachandran plot. In our previous video we have already discussed about amino acids. Now we know that the general structure of amino acid, it contains alpha carboxyl group, alpha amino group, R side chain, each atom attached to alpha carbon. Now what is peptide? Peptides are molecules that are formed when two or more than 50 amino acid residues are joined by a bond called peptide bond. Now you see here in this structure, this is first amino acid residue, here it is second amino acid residue, and here it is third amino acid residue. Now the molecule formed here is known as peptide. Now since here three amino acid residues are joined, therefore it is known as tripeptide. Similarly, when two amino acid residues are joined, it will form dipeptide, and when more than 50 amino acid residues are joined, it will form polypeptide. Now you see here the amino acid residue containing amino group will be known as N-terminal amino acid residue, and the amino acid residue containing carboxyl group will be known as C-terminal amino acid residue. C for carboxyl group and N for amino group. So peptides are molecules consisting of 2 to 50 amino acid residue covalently linked by a bond called peptide bond. So now we know that amino acid residue, when joined by a peptide bond, they form peptide. Now what is peptide bond? So peptide bond are amide linkages formed between the alpha carboxyl group of one amino acid residue and the alpha amino group of another amino acid residue. And the reaction here involved is known as dehydration reaction. Here you see, this is first amino acid residue and this is second amino acid residue. The OH of the alpha carboxyl group of first amino acid residue and the H of the alpha amino group of second amino acid residue will be removed as water molecule H2O, and there will be a bond formation between the alpha carboxyl group of one amino acid residue and the alpha amino group of another amino acid residue, and the bond formed here is known as peptide bond. And since here in this reaction H2O is removed, water molecule is removed, therefore this reaction involved is dehydration reaction. Now you see that peptide bond has some characteristics. Here this point says the peptide bond has a partial double bond character, which means the bond formed between the alpha carboxyl group and the alpha amino group, although it seems to be pure single bond, but actually it acts as a double bond. Therefore peptide bond has a partial double bond character. Next point says the partial double bond character keeps the peptide bond in a rigid planar configuration. Now you see, when a pair of amino acid residues are joined by a peptide bond, 6 atoms of the peptide group, that is carbon, nitrogen, hydrogen, oxygen, C alpha and another C alpha, these 6 atoms will be on the same plane. You see here in this structure, this is peptide bond, the CO and C alpha of first amino acid residue and NH and C alpha of second amino acid residue are all on the same plane. Therefore, peptide bond has a rigid planar configuration. Here this point says due to partial double bond character, peptide bond has two configurations. One is cis configuration and the other is trans configuration. In cis configuration, you will see that two successive alpha carbon atoms will be on the same side of the peptide bond. Here it is the peptide bond and the successive alpha carbon will be on the same side. While in trans configuration, the successive alpha carbon atom will be on the opposite side of the peptide bond. You see here in this figure, in trans configuration, the successive alpha carbon atom is on the opposite side of the peptide bond, while in cis configuration, the successive alpha carbon atom are on the same side of the peptide bond. And you will see mostly all peptides are in trans configuration because of less steric interference. Next point says the restricted rotation CN bond can be specified by torsion angle omega. Now we know that the bond between the alpha carboxyl group and the alpha amino group has a partial double bond character, and because of this partial double bond character it is restricted to rotation, that is it cannot rotate, and this restricted rotation is specified by torsion angle omega. So these are few characteristics of peptide bond. So what we have discussed about peptide bond is peptide bond has a partial double bond character. And due to partial double bond character, it has a rigid planar configuration. And due to partial double bond character, it also exists in cis and trans configuration. And the restricted rotation is specified by torsion angle omega. So these are few characteristics of peptide bond. Now we know that the bond between the alpha carboxyl group and the alpha amino group is known as the peptide bond. But apart from this peptide bond, there are also two bonds present, that is between the amino nitrogen and the alpha carbon and the alpha carbon and the carbonyl carbon. So here this point says the bonds between the amino nitrogen and the alpha carbon atom and the alpha carbon and the carbonyl carbon are pure single bond. That is, the bond form here is pure single bond. It will not have a partial double bond character as the peptide bond. The next point says the rotation between the bonds N and C alpha and C alpha and C can be specified by torsion angle phi and psi respectively. That is, the bond formed here is, since it is a single bond, they are allowed for rotation, and the rotation between the N and C alpha is specified by torsion angle phi, whereas the rotation between the C alpha and C is specified by torsion angle psi. Next point says phi and psi can have value between plus 180 degree and minus 180 degree, that is 360 degree for each. And the next point here says most value of phi and psi are not allowed due to steric interference. So the phi and psi can have value ranging from plus 180 degree to minus 180 degree, that is 360 degree for each. But most of the value of phi and psi are not allowed due to steric interference. So you see, when we place atoms in three-dimensional structure, we see that atoms can take up space. And now two atoms will not able to take the same place at the same time, and there will be a clash between the atoms, and this physical clash between the atoms is known as steric interference. And because of this, most value of phi and psi are not allowed, and only few values are allowed due to steric interference. Now we know that most value of phi and psi are not allowed due to steric interference. Now there was one scientist named G.N. Ramachandran who first discovered the combination of phi and psi value that were allowed and that were not allowed due to steric interference. So Ramachandran plot is a two-dimensional plot where permitted value of phi and psi are visualized. Now you see here in this figure, the darker blue region is for the allowed values of phi and psi, whereas this lighter blue region is for the restricted value of phi and psi. Now there are some amino acids like glycine. Glycine, since we know that it is the simplest amino acid residue because it has each atom as its side chain, therefore it has a less steric interference, and because of less steric interference it is considered to have broader allowed region. Whereas proline, since it has a ring-like structure on its side chain, therefore it is considered to be conformationally most restricted amino acid. So what we have discussed is that Ramachandran plot is a two-dimensional plot where permitted values of phi and psi are visualized, and it was first discovered by the scientist named G.N. Ramachandran. So that's all for this video, and if this video was helpful for you all, please like it and subscribe it. Thank you.